Bismillahir Rahmanir Rahim. Inflammatory memory is the idea that tissues and cells can remember past inflammatory events and respond more vigorously or abnormally to subsequent challenges. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University, and our video today is about inflammatory memory and chronic dermatosis. Although adaptive immune cells, such as T cells, have long been recognized as key players in maintaining disease memory in chronic dermatosis like psoriasis, the memory is not exclusive to the adaptive immune system. Instead, non-specific forms of training and imprinting occur across a range of skin-resident cells, including innate immune cells, epithelial stem cells, and possibly other non-immune cells. For example, psoriasis, a prototypical chronic relapsing inflammatory skin disease, has traditionally been viewed through the lens of adaptive immunity. Tissue-resident memory T cells, particularly those producing interleukin-17, remain in previously affected skin long after clinical resolution. Their presence can drive location-specific relapse, suggesting that post-lesional skin retains immunological imprints of disease that predispose it to future flares. However, beyond T cells, innate immune cells can also be epigenetically trained by inflammatory cells. This training, originally documented in contexts like fungal infection and lipopolysaccharide exposure, results in long-lived epigenetic modification that reprogram progenitor cells in the bone marrow, as well as tissue-resident macrophages and other innate cells. Such changes can either heighten or dampen responses upon re-exposure to stressors or pathogens. Although this phenomenon is well characterized in some systems, its precise role in chronic skin disease remains to be fully understood. Recent studies show that skin epithelial stem cells and potentially other stromal cells like fibroblasts and endothelial cells also remember past inflammation. After an inflammatory insult, epithelial stem cells undergo lasting epigenetic modifications that changes in chromatin accessibility, histone marks, and persistent binding of transcription factors. Even the skin appears clinically normal, these molecular marks prime the cells for rapid and exaggerated responses to subsequent injuries or stress. Such memory can enhance wound healing but can also predispose tissue to maladaptive inflammatory actions or neoplastic transformation over time. Inflammatory memory is largely governed by epigenetic regulation, persistent histone modifications, DNA methylation patterns, accessible chromatin regions, non-coding RNAs, and transcription factor bookmarking collectively encode long-term changes in gene responsiveness. The induction of trained immunity involves metabolic and epigenetic reprogramming in both circulating myeloid cells and bone marrow progenitors. These changes lead to long-term functional alterations, enhancing the body's ability to combat infections. These non-genetic modifications allow cells to quickly recall past states without altering their underlying DNA sequence, enabling rapid reactivation of key inflammatory or stress response genes upon secondary stimuli. Understanding these layered mechanisms of inflammatory memory has significant clinical implications. It challenges the traditional focus on adaptive immunity alone and opens possibilities for new treatments targeting the memory state in various cell populations. Erasing or modulating these epigenetic imprints could potentially prevent relapses and lead to more durable remissions in psoriasis and other inflammatory skin disorders. Such strategies might shift the current paradigm from continuously suppressing inflammation to fundamentally reprogramming the tissues to become less reactive. In psoriasis, skin lesions often reappear at previously affected sites after systemic therapies are discontinued. Despite initial clinical remission, the skin retains long-lived molecular and cellular signatures of past inflammation, which can predispose it to flare when exposed to even minor triggers. So, at the heart of this molecular scar is a multifaceted interplay of immunologic and structural factors. Tissue-resident memory T cells, particularly interleukin-17 producing CD8 positive tissue-resident memory cells, persist within the epidermis of resolved psoriatic plaques. These T cells maintain a heightened capacity for rapid reactivation, possibly facilitated by local antigen presentation and sustained interleukin-23 signaling. Similarly, Langerhans cells and dermal dendritic cells in clinically clear skin remain primed to produce pro-inflammatory cytokines. Non-immune cell populations, including keratinocytes and fibroblasts, also show epigenetic adjustments that leave them ready to overreact to subsequent stimuli. Abnormal vascular and lymphatic architecture further contributes to a pool conducive to inflammation's return. Collectively, alterations in DNA accessibility, transcription factor binding, and regulatory RNA networks, along with persistently restructured tissue landscapes, create a memory that stays long after visible symptoms subside. As a conclusion, chronic inflammatory skin diseases like psoriasis are shaped by a complex interplay of adaptive and innate immune memory as well as epigenetic imprinting in non-immune cells. This broadened perspective on disease memory points towards novel approaches aimed at targeting these persistent cellular and molecular changes to achieve lasting remission and tissue health. Lastly, I hope that you have enjoyed this video, and again I'm reminding you to subscribe to the channel and activating the alarming bell, and thank you.